Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
Scott, how's it going?
Great.
Did you end up having turkey at your friend's place last week? You weren't quite sure about the menu last week.
Well, we did indeed, and it was turkey, and he's a fantastic, he's a chef, so yeah, it was great.
Did you brine it or just...
Actually, I think he brines it.
Okay.
That's a good point.
We did that this year, and what a difference, an amazing difference.
I know, eh? But he's a chef. He's not a fancy chef. He worked at a ship, But the great thing about that is he's a meat and potatoes, but he knows how to do everything. It's amazing how even the simple things like mashed potatoes, you can do that really well, or you can kind of do it so-so. In my case, it's usually so-so. But everything he did was just that little bit of extra, and the stuffing was the best stuffing I've ever had. So it was great.
All right, we're getting the listeners hungry.
Yeah.
And you had a good Thanksgiving break as well.
Yeah, very nice.
Good. And the episode,
How about you?
We actually did our Thanksgiving the week before. We were up north the week before, so last week was really a quiet one for my wife and I.
That's nice to do it up north. I've always wanted to do that. I've never been able to, but yeah.
Yeah. Now, the interview we did last week, quite a bit of science there, eh?
Yeah, a lot of science.
Maybe too much.
As you know, I have a propensity for, but maybe a bit much for some, yeah.
Yeah. Okay. So I want to explain something to the listeners here. I have been getting some feedback over the past couple months. The episodes have been getting pretty far into the deep end of the scientific weeds. I've never had to drop in so many of those little sound booth inserts explaining a particular concept or a word as I have over the past couple months. And I've even had one listener very recently question whether this is a religious podcast anymore. He said it's basically a science podcast now.
Yeah, well, I get the question. I can see why somebody might say. In fact, you and I have had that discussion privately. Right. So, yeah.
Okay, so here's my disclaimer then. We did make the decision to take a deep dive into intelligent design, which, despite what they say, is very much a religious worldview. Instead, they would claim it's an alternative science or a scientific worldview, and much of their arguments are rooted in science. And so we've had to fight that battle on their home turf. But today will be the last science-heavy episode for a while. We're going to take a break from the deep end of science and get back into the social issues and the theological, religious, and philosophical issues that we've often done.
For sure. And I think having presented some of this background is going to be helpful to people as we do move forward and talk about some of those theological, philosophical type issues. So I think it lays a good foundation for those discussions.
Right. Okay, so last week we took a close look at this long-term evolution experiment. We got halfway through that interview with Dr. Zachary Blount. He told us quite a bit about the project, including the history. It began with a visionary scientist, his mentor, 40 years ago, with the goal of probing how evolution works. And that study gained a lot of international recognition for the data and insights it started generating. But this groundbreaking project has been panned by creationists and ID proponents alike for showing exactly how evolution doesn't work, or at least that's how they see it. But after hearing from one of the members of the long-term evolution experiment, Dr. Zachary Blount, last week, one of the many people doing the work and collecting the data, analyzing and directly interpreting the results, I don't know about you, Scott, but I come away with a very firm conclusion that those creationists and ID proponents don't have the necessary direct hands-on experience with the science. Most of them are just very clearly misinformed and totally inadequate to be an authority on the subject that they're claiming to be. And even worse, some of them seem to be intentionally distorting the data and deceiving their listeners.
Yeah, it would certainly seem that way after listening to, really both sides of the issue. And I just kind of, as you're talking, I'm kind of jokingly thinking somebody's saying, well, after 50,000 generations, it hasn't turned into a fish.
Yeah, I know. Yeah. Well, and that was the question he essentially asked. They didn't show whole new nanomachines, and we talked about it. Oh, that was really a bit of a stretch.
Yeah.
But, in the course of tracking 76,000 generations of bacteria, which is the human equivalent of 1.5 million years of human evolution, the LTEE, this long-term evolution experiment, have already found dozens of changes in 57 different genes. And the most commonly heard criticism from creationists and ID proponents alike about the LTEE is that it only showed evolution proceeding through the breaking of genes and decreasing the amount of information in a genome. Now, to be fair, in some cases, some of the genes have indeed been turned off or damaged. This is what humans do when we train for a marathon. We lose fat. We reduce the weight of our clothing, especially clothes. We stop eating certain foods. And this is also what happens all the time out in the wild. Evolution does exactly this. Our own bodies are full of such broken genes involved in taste and smell, involved in making vitamin C. In some cases, those broken genes and functions are part of the story of human evolution and divergence from our pre-hominid ancestors. Scott, did you know that we humans have an inactivated gene that's responsible for humans no longer having a tail, and that some people, in rare cases, are born with a tail that has to be surgically removed?
That I hadn't heard of. No, that's interesting. I had no idea that people could still be born with tails.
It's very rare, to be fair,
sure,
but the fact
sure.
that it happens, and there's a number of ways in which it happens, but one of which is it has to do with the genes that are involved in forming and deforming, removing the vertebra. You know,
Right.
as the spine develops, it has to be stopped at some point, and in these cases, it just keeps on going. So there are genes that get turned off and inactivated and broken, but in a lot of other cases, what the most important pathway is by a new gene, a new function arising through the duplication of genes, followed by a series of changes to one of the copies. A very narrow perspective on those changes is that the original function is now broken, in quotes. But a more nuanced perspective sees this as setting the stage for new innovations. Half the technical innovations and inventions made by humans came about by making something less able to do its original function and making it better able to do something new. That's how humans turned the first combustion engine automobile into a lawnmower or a motorboat. How we turned radar transmitters into microwave ovens and turned calculators into word processors. All of those innovations involve taking existing technology, existing information, and redirecting it to a new purpose.
Yeah, it's interesting. And this gets into a conundrum that we're going to talk about in the future episode. Because when we say innovation, of course, that's a very human thing, right? We think of innovation, we automatically think of innovative design, right? But we're not talking about that. What we're talking about is the fact that through these mutations, things get repurposed by natural selection. It's not that they were innovated in a way that somebody, like the way that we might innovate, they look at some parts and think, I can use this a different way. No, what happens is these parts exist, and then there's some sort of environmental pressure or some sort of, and guess what? It works. It happens to work. So I just want to be clear, because I know one of the things that critics will often say is, yeah, you guys, you say you don't believe in design, but you're constantly talking about design. But we're not, and we are going to take that up at another time
next week.
Next week. But I do think it's important to point out, even as we have this discussion.
Now, getting more to the subject of this episode, those creationists and ID proponents will almost always point to what they see as the prime example of breaking genes and degrading genetic information. They'll point to the LTEE's discovery of changes to a gene involved in the metabolism of an energy substrate called citrate. But both of those details will be wrong. The citrate gene was not broken, and the LTE showed evolution occurring through the building up of genetic information, new genes, and new functions. So in today's episode, we're going to hear about how a copy of a particular gene was inserted into a place in the genome where it could become active under conditions where it normally was not active. That does not mean broken. And let me use a common household example of what I mean by that to help the listener see that I'm not just playing games with delicate wordplay here. Imagine a light controlled by a light switch. The switch is strategically placed at a door leading into a very large room which has several doors leading into it. Ideally, you don't want to always have to walk to the far end of the room just to turn on that light. Or a more common example of what I'm talking about here would be the light switch at the door leading to a dark basement. You turn on the basement light and go down the stairs, but then someone turns the light switch off and you're left completely in the dark. So in both of those cases, you'll find a second light switch that allows you to turn the light on when the first switch has it turned off. Scott, do you have any of those kinds of light switch situations in your house?
Yeah, well, in our case, actually, we don't have a basement, but it's a 2-story. And yeah, there's a light at the bottom of the stairs, and there's a switch at the top of the stairs. And if you go to bed, you want the light on while you're going up the stairs. But when you get up the stairs, of course, you don't want the light on.
Exactly.
Right? So it makes total sense. Yep.
Right. And so that's not an example of breaking the light. It's not an example of breaking the light circuit, but simply an example of adding a second control point. And so we're going to hear in today's episode that citrate metabolism is normally turned off in the presence of oxygen, but that a second control circuit is inserted so that it can be turned on in the presence of oxygen. Not breaking it, just giving it another control. So today we're going to focus particularly on that citrate metabolism story because, again, that's the story that keeps getting brought up by creationists and ID proponents who just keep getting that story all wrong, unintentionally or otherwise. But first, I need to get the listeners ready for this. I already mentioned last week and again when we started this episode that this is going to be the most science-heavy podcast we've ever done. And most of that high-level science will be put on the table today. So I want to first give the listeners a mental image and some background information to help them keep up with the science here. And if the listener really, really wants to get nerdy and get a solid grasp on what we're talking about here, I'd recommend they go to the episode show notes, which will give them an actual picture that puts some of the important details into perspective. They can keep this open while Dr. Blount unpacks the citrate metabolism story, and I'm convinced it will help them put all the pieces into place. So you might want to pause the episode at this point and get that graphic on your screen. Just go to the show notes and there will be a link and just open it up. My apologies to those of you who tend to listen to us while you're commuting in a car or jogging. Unfortunately, you're going to have to just do this in your mind. So the first conceptual image that I want to put in the listener's mind is one that I actually did use last week. Building a campfire either with solid oak logs or with twigs and leaves that you find scattered on the forest floor. The oak logs are going to produce a much better fire, a lot more light and heat, and a lot less smoke than the twigs and leaves. But as I said last week, when you're freezing in the forest, you'll do whatever it takes to get heat and light, even if that means trying to burn twigs and leaves. So in the graphic that I created for the listeners, you'll see a series of buckets with glucose flowing in through the top and producing a lot of energy molecules, so the little E's in the figure, and then citrate flowing in closer to the bottom and producing only a little bit of energy molecules. I think that much you can get from the figure without me saying much more, but I will explain the figure in a lot more detail in a couple minutes. But what I've shown you there is the bacteria trying to get energy from two different fuel sources. Glucose, which is the equivalent of the oak logs, and citrate, the equivalent of the twigs and leaves. So the bacterium has all the genetic machinery that it needs to burn glucose, and what that machinery does is convert the glucose through a series of biochemical steps into other molecules. This is just like your car engine converting gasoline into other molecules. You probably know them: carbon dioxide, water, and a lot of exhaust fumes. I'd like you to imagine this bacterial metabolism like a series of buckets. Notice that some of the buckets have large drain holes. Whatever's flowing into it flows out very quickly and generates a lot of energy, all those little E's. But then a series of other buckets that have small drain holes, generating a little bit of energy. And at each step, as the molecules flow from the one bucket to the other, that represents a biochemical conversion from one molecule type to another molecule type. And the names of those buckets and the names of those molecules aren't important, with a few exceptions. I want you to notice that the one coming in at the top is glucose. It's flowing in at the top, pouring all the way through, generating electricity as it goes to the next bucket and the next bucket. Eventually it becomes a molecule called citrate. And we're going to hear in a few moments that citrate comes from the molecule beforehand, which is acetate, but I'm going to put that on pause for the moment. Glucose flows through these buckets and becomes citrate and produces a lot of energy in the process. And then the citrate continues on down and creates a little bit of energy. And again, that was the oak logs at the top and the twigs at the bottom. So I think at this point, I'm going to stop there, and I want to emphasize I'm really simplifying things. So the biochemists in the audience, which includes our guest speaker, Zach, I'm sure he's listening, they're all probably squirming in the seats right now because they want all the details, and I'm keeping the details to a bare minimum. But is this much clear, Scott?
Oh, yeah, for sure. The goal here is not to, of course, in microbiology, the goal is to give people kind of an overall understanding of the evolutionary process at a higher level. And I realize, the devil or the science is in the details.
Exactly.
But we're not doing research, but we can certainly we can understand how this process works, at that higher level.
Right. And I think where this graphic will become useful to listeners here now is that Zachary will make mention about citrate, which will come up quite a bit, but he'll occasionally mention succinate and acetate. And just to know where these are fitting together and how they correspond to each other, I think that's where this graphic will help you to put all the pieces in place.
Yeah.
So at this stage, I want to put together these two metaphors that I used. The metaphor of the two light switches supplying electricity to the same light, and this image of glucose and citrate making their way through these series of buckets and generating energy. And I want to put those two together to show how the mutated cells that Zach studied outcompeted their non-mutated cousins by acquiring the ability to grow on citrate. And again, this is to help clear up the misunderstanding that keeps coming up from creationists and ID proponents. So here's my thumbnail sketch of the story, and when you hear Zach tell the story, you'll be able to better follow along and put the details in place in this graphic that I've given you. So the story begins with the fact that the E. coli bugs normally don't grow on citrate in the presence of oxygen. In my analogy of a large room with the two doors on opposite sides and only one light switch at the one door, the large room is a non-mutated bacterium. The light switch is a trap door in the floor that lets citrate into the room. Zachary will call that the citrate transporter, or CIT-T, and the one light switch is an oxygen sensor. That trap door only opens when there's no oxygen in the room. Thing is, the normal conditions of the lab is that there's always oxygen around. These lab guys are not wearing spacesuits with oxygen masks. They're just working in normal environments, so oxygen is always around, so that switch never gets flipped on in the bacterium. That trapdoor doesn't open in the non-mutated bacterium. They can still grow perfectly fine on glucose using the oxygen, but when the glucose starts running out because the bugs are multiplying like crazy and using up all the glucose does run out and the bugs just simply stop growing. The mutated bugs, on the other hand, have added a second electrical switch. They've left the first electrical switch intact. They haven't changed it in any way. It's not broken. And so that first switch still fails to open the trapdoor when oxygen is present, although it would still open the door if oxygen were removed, but again, the researchers don't do that. But this second electrical switch that the mutated bugs have added in isn't regulated by the presence or absence of oxygen. Instead, it's regulated by the presence or absence of glucose. So they grow on the glucose along with their non-mutated cousins. But when the glucose levels drop and their cousins start slowing down, these mutated bugs have that second electrical switch that now gets flipped because glucose levels are dropping, and the second switch opens the trapdoor and the mutated bugs start using citrate for energy. Like the Energizer bunny, they just keep right on going while their non-mutated cousins all shut down because the glucose levels are dropping. And so over the course of the experiment, the mutant bugs eventually outnumber the non-mutant ones. The latter get crowded out and eventually go extinct while the mutated bugs continue merrily along with this new function. The population has now evolved into a new kind of E. coli. And again, because it needs to be emphasized so clearly to the Ken Hans and Stephen Myers in this discussion, those bugs have evolved not by breaking a gene, a regulatory pathway, that switch that is regulated by oxygen levels, but instead by making a copy and inserting that into a new position in the genome, a position that is regulated by glucose levels, creating a new regulatory pathway.
Yeah, wow. It's just kind of mind-boggling, it really is fascinating.
That the bacterium can do that, or that the creationists can get it all distorted?
Well, a little bit of both. A little bit of both. Or maybe a lot of both. As to the issue of the design folks and creationists and so on, as to why, I think we know why, the motivations, but it's a shame that they're misrepresent the case, even though it's been well documented and explained, which is, I think, obviously it's a deeper issue, and it's why in general that movement is so opposed by the scientific community, because they say, well, you don't have a scientific mindset here, because a scientific mindset says, if I have a hypothesis and the data comes in and it's tending away from the hypothesis or even falsifies it, well, then I move on,
Right.
And in fact, a really good scientist is actually kind of excited when a hypothesis breaks, because that probably means they're on the precipice of something really interesting.
Right,
Something,
exactly.
yeah, some new vista of knowledge. So we shouldn't be afraid of that. We should, in fact, embrace it. But yeah, how this comes together and it creates this dual kind of system, it is fascinating. It brings up a lot of questions that maybe we can talk about a little bit later on, in my mind anyway.
Right.
Yeah, keep going.
Okay, so this is one of the lessons that we learned about evolution, that evolution is often about making duplicate copies of genes and modifying the duplicates so that those duplicates can now do new things. It's like disconnecting a car engine from the car's wheels, turning it sideways and connecting it to a spinning blade, and now you've got a lawnmower instead of a car. A new function is created by taking pieces of the old information, changing them a bit, and making something new. Now, Zach will also tell us that the evolution of this new citrate metabolism function in his E. coli bugs, it followed a pattern that's often seen in evolution, that it often happens, and it happened here, in 3 steps. Those 3 steps are really very easy to understand. The only problem for lay people is that scientists like Zach will use big, long, fancy words to name them. So basically the 3 steps are these. First, you prepare for the new function. Second, you get the new function, often in a very crude and weak and inefficient form. And then 3, you refine that new function to make it more efficient and strong. So again, prepare, get, and then refine. Except Zachary will refer to these as, well, the preparing would be potentiation, getting it would be actualization or instantiation, and refining it would be, well, refinement. And we see those same 3 steps in the human world of inventions and discoveries. Powered air flight was actualized, instantiated in 1903 by the Wright brothers. And it was a very measly flight that only lasted 12 seconds, covered only a couple hundred feet at a height of, at best, 10 feet off the ground. But within a few decades, that new function that had just been instantiated was refined, so that people are now flying around the world at thousands of feet above the ground. And that new function first required a number of other developments that had nothing to do with flight. The development of the combustion engine and the propeller and making strong structures out of balsa wood. Those 3 things that I just mentioned and many others had to be in place before the first flight could be actualized. So they potentiated the function of flight. And there again you see those 3 steps: prepare, get, and refine. Potentiation, actualization, and refinement.
And right now I can just hear the ID folks saying, yeah, exactly. That's what humans do. They design things. And that's how you design things. And if nature works that way, then guess what? There must be a designer. Well, the difference is, what we're saying is, yes, that process does occur, but it occurs through this mechanism of random mutation and selection. And
right.
more than that, I think now with epigenetics and we're learning all the time about other mechanisms that are playing into this process. So I don't think anybody believes we're done with understanding evolution. We're learning all the time there are other forces at work, even, that we don't yet fully understand. But the fact is that even what we do know demonstrates that innovations and novelty can occur in
right.
nature through this process. Yeah. It's interesting. My background's my career is in technology, particularly software development. And any of our listeners that are involved in technology or software development in particular will know about what we call the iterative process, right? And in an iterative process, we do a little bit of work, we create something, and then we test that thing, and then we break it, and then we refine it, and then we do a little more work, we test, refine. That's what you've described is exactly how we develop in the software and hardware and many other things.
Right. Okay, so getting back to citrate metabolism in E. coli, the second step in that 3-step process, the actualization step, was, and we're going to hear this from Zach, was the actual insertion of a copy of the citrate transporter gene into a new position in the genome, one that put it under the regulation of glucose levels. And when that happened at generation 30,000, roughly, and so we'll call those the Gen 30K bugs, the LTEE scientists found their cultures were all cloudy and the mutants had taken over. That was the actualization of the new function, but that actualization required a number of other potentiating steps, mutations that occurred long before. if I have the story right, and Zach will tell us more specifically, there was an important mutation that related to acetate metabolism roughly at 10,000 generations, and then another change at 20,000 generations. So we'll call those the Gen 10K bugs and the Gen 20K bugs. And this is where the story of the mutant bacteria gets particularly complicated. So hopefully Scott and I can give the listeners enough of a framework with this graphic to help them digest what Zach's about to tell them.
What you're describing here, it looks, here's an analogy for you. It looks like an overnight star, right? They'll talk about how people became a star overnight. There's almost no examples in the real world of people becoming a star overnight. If you look at anybody, you'll find that there was tons of things beforehand that led up to that.
Set the stage.
But it looks like on the surface, wow, they were born a star. They were a star overnight, and it reminds me of that.
Yeah. So let's go back to that graphic and my analogy of making a fire from oak logs versus twigs and leaves. You can get some heat from the twigs and leaves, certainly not as much from the logs. And in the same way, the E. coli bugs can get some energy from citrate. Opening up the spigot on the bucket for citrate and letting it make its way through those series of buckets and biochemical processes. And just to help make the point, let's just say that the glucose makes 10 energy molecules, but citrate only makes 2 or 3 energy molecules. And it takes time for the glucose to make its way slowly through those buckets, being broken down into other kinds of molecules and in the process generating 10 energy molecules. One of those breakdown products of glucose is a molecule called acetate. One of the buckets contains the acetate. The sugar called glucose gets broken down to the acid in vinegar, acetate, and eventually to the acid you find in oranges, citrate. But long before the Gen 30K bugs learned about the trap door for citrate that we talked about, the room with the 2 switches, some of their ancestors, the Gen 10K bugs, learned about a shortcut, that it was possible to create another door for acetate, a trap door for acetate, and let acetate drop out of the process of metabolizing glucose. So those Gen 10K bugs would only get 6 or 7 energy molecules from glucose instead of 10, which sounds like a bad idea, except that the first half of the glucose metabolism pathway happens really fast while the second one happens a lot more slowly, such that the whole rate of metabolizing the glucose completely gets slowed down by that second half. So the Gen 10K bugs realize they can metabolize a lot more glucose and get a lot more energy by just getting the first half of the energy really, really fast and throwing away the half-digested glucose in the form of acetate to make room for some new undigested glucose. Why spend an hour getting 100% of the energy out of one molecule of glucose if you can extract half the energy out of 10 molecules in the same amount of time? It's very inefficient, but 5 times more productive, which means 5 times as much growth and as much reproduction. So these Gen 10K bugs started spinning their tires, burning gallons of glucose and spewing acetate everywhere, outcompeting the slower, more efficient bugs, and won the race of survival. So it's no different than the hotshot kid who likes to put the pedal to the metal everywhere he drives, burning up a lot more gas than grandpa, but getting there a lot faster. Wasteful, absolutely, but when your only measuring stick is getting to the destination, then wasteful is better.
I think this is an important point, because one of the things that I thought as Zach was describing this generational thing and the fact that there were the number of precursors before we ended up with the citrate-loving E. coli, one of my questions was, but why did those things survive? So that's what you're explaining, that for that purpose, it did offer an advantage, and the bug took advantage of that, reproduced more and so on. That's how it was retained.
Right.
And then eventually there were other things. It's kind of a convergence of things. Yeah. But so each step along the way, let me ask that question. Does each step along the way have to offer some advantage to the organism, or could it just be neutral, tolerated by the organism, but just sit there, and then sometime later something comes along that makes use of it? Is that possible?
Well, some of the changes will be negligible, will have no real effect. Some will have positive effect, and some will be negative. A lot of them will be negative, and they found 57 genetic changes, but those were the successful, the ones that gave a benefit, that survived. I'm taking a guess here, there could have been 1,000 or 1,000,000 genetic changes, most of which were not good, but these 57 actually made it better.
yeah, I think that's interesting that there can be a lot of genetic changes that are just neutral. They don't confer a benefit necessarily, but they're sitting there.
Right.
And that just adds to the possibility for evolution at a later time. So in other words, it's not just you get the deleterious changes that actually might, they're not going to help. And you've got the ones that do confer an advantage. So they're going to help. And then you've got the neutral ones, but there's probably a lot of neutral ones that later confer an advantage.
Yeah.
I would think. I don't know if we have examples of that, but I guess what I'm trying to say is 2 out of the 3 types of mutations are useful to evolution. The only one that really isn't, I guess, would be the deleterious one.
Right. So the Gen 10K bugs developed a trapdoor for acetate, which let them grow faster, but at the cost of eating the glucose a lot less efficiently and fouling up the atmosphere with an acetate smog. Acetate is toxic, which otherwise might begin to kill off the bugs, but they developed other mutations that allowed them to use the acetate as another food source. And here's the point. Some of those mutations enable the bugs to turn the acetate into citrate and develop the ability to use citrate as another food source. And so this becomes the potentiation part of the story. The Gen 10K bugs became acetate eaters, which then set the stage for the actualization part of the citrate story. The bugs becoming citrate users, citrate eaters at Gen 20K. And then that new function became further refined until when we get to the Gen 30K bugs, the researchers discovered this new kind of bug turning their test tubes all cloudy. The bugs had become superbugs and almost wiped out their non-mutant cousins.
Yeah. So we may have evolved a new bug.
Maybe, yeah.
I just curious. Yeah. Okay.
Yeah. So let's draw the line there and let Zach now tell the citrate story, and we'll come back when he's done and pick at some of the loose threads at that point. The thing that creationists and ID proponents will often say. You did mention citrate, and that's the one in particular they always focus on. So maybe, Zach, tell us a bit more about this whole citrate story again.
Okay. So as I noted earlier, the citrate's been the medium that we use in the experiment since the very beginning, but E. coli cannot ordinarily make use of it as a food resource when oxygen is present as it is in the lab. But after about 30,000 generations, in 16 years of the experiment, one of the populations evolved the ability to do so. This is a population called Era-3. This is the one that was noted by Tim Cooper when he came in to do the transfers one day, and one of the populations was cloudier with more bacteria. So, as I said then, it was discovered that this population was cloudier because it was full of variant bacteria that had evolved in the population that were able to eat citrate. And we know that these spontaneously evolved from the populations because by that point, we knew of these other mutational changes that had taken place. So like the PYKF mutation that I mentioned, there is one of those in the Era-3 population, and we knew what that mutation was. So we were able to sequence that gene in these citrate users and see that it had the mutation that marked them as a part of this population. So we had this novel trait in the context of this organism, in the context of this experiment, and it had only happened once, and that opened all sorts of questions. Now, before I go on to that, I just want to note that it's often represented that these...
Can I interrupt for a second? You said it only happened once. Do you mean it happened once in one of the 12 populations that your lab are growing?
Yes. So the citrate users, what we call CIT+, have only evolved in one of the 12 populations.
Okay.
And that's true even until today. So it has not happened again. And I'll get into a question that poses in just a moment. I just want to mention that something that I found out when I went to study this population, I went into the what we call the frozen fossil record of all of those archived frozen time points for this population. And the funny thing about bacteria is, if you freeze a zebra or a human, you get a dead zebra and a manslaughter charge if you warm them up. It's not like that with E. coli. So you freeze E. coli, you warm it up, it's fine, it starts growing again. So this frozen fossil record is of viable cells. So we have, go ahead.
And again, I just want to help the listeners. I know you said this, but I want to remind them, Your team goes into the lab every day and just moves over 1% of the bacteria. You throw away the 99 usually, but every 500th time you hang on to that 99%, stick it in the freezer, and then you carry on and rinse and repeat.
Right. Every 500 generations, which is
Right.
is every 75 days. Every 75 days, we freeze
The
what
99%.
remains of the population. Exactly.
Right.
And that becomes a new layer in this frozen fossil record. Viable cells that will remain viable for the foreseeable future until background radiation shreds their genomes millions of years from now, assuming the power stays on. So I went back into the frozen fossil record of this population to see when the CIP plus guys arose. And it turned out that they had arisen a year before. But number one, they were very bad at using citrate, and they were very rare in the population. And the thing is, even though they were very poor at using citrate, once those CIP plus guys were around, they were visible to natural selection. New variants arose. Some of those rare mutations improved their ability to grow in citrate. So the trait was improved until shortly after 33,000 generations, there were CIP plus variants that were strong enough at using the citrate under the conditions of the experiment that they rose to become dominant. And that's when we noticed them. So there's that. And that opens a question too that I'll get to. So one of the dominant questions we have with the citrate users is, again, they arose only once in the 12 populations over the course of the entire experiment, even though the citrate was always present. So when I came to the lab, one of the questions that I was focused on and that has gone on to guide me in my subsequent work was the question, how come? How come it only occurred once? And there were two possibilities. One was that it was a single rare mutation of really big effect that could have occurred in any of the 12 populations at any time. And when it occurred, it would allow growth on citrate. The other goes to what's called historical contingency. So historical contingency is this principle that in historical systems, different histories can lead to different outcomes. We'll talk more about historical contingency later, but that's kind of the key idea here, that particular histories lead to particular outcomes. So it's possible that the CIP plus evolution event was a particular outcome that depended on the particular history of this population. And so what I posited was that this population had been special because over the course of its history, one or more mutations arose that made it easier to evolve CIP plus. I call this potentiation. These mutations potentiated the evolution of CIP plus. And this was a really remarkable question. It goes to some stuff that Stephen G. Gould, the late evolutionary biologist and paleontologist and writer, talked about a lot. And we had this opportunity to test contingency. And we were able to do it by making real a thought experiment that Gould had written about called replaying life's tape. He talked about in his book, Wonderful Life, this notion that if you were to go back to different points in the distant past and start evolution over again, that would you see the same living world evolve, or would it be completely different each time because history was? And when he wrote about that experiment, he said, the unfortunate thing is it's impossible. You can't do it. But we could actually do it in this experiment.
Right, yeah.
Yeah, because we have that frozen fossil record. And so I was able to go back into the fossil record at different time points and take out CIP minus cells that were unable to grow in citrate and then re-evolve them and start evolution over again to see if the CIP plus mutation would occur again or event would occur again. And so I did this under a number of different conditions over the course of about 2.5 years, replaying the tape of evolution to see how often CIP plus arose, because the two possibilities, rare mutation and contingency, made different predictions. If it were a rare mutation, then we would expect in these replay experiments that CIP plus could arise in any of them, regardless of when it was started. Whereas if it were contingency, And because of potentiating mutations that have arisen in this population, then we would expect to see more events of re-evolution of CIP+ in populations that were started in later time, after those potentiating mutations were in place. And that latter possibility is exactly what we saw. I ended up going through and testing, I think it was about 44.4 trillion cells.
Wow.
Over the course. It was a lot of work. I was in the lab a lot. And then I ended up seeing, I think, 21 instances of re-evolution of CIP+. And in every single case, it was from a population that was founded from a clone from generation 20,000 or later.
Okay.
So this demonstrated that this was a historically contingent trait. It was the first time that a historically contingent trait had been demonstrated to be historically contingent by experiment. And then that brings up the question of what was it contingent on? What were those mutations?
Right.
And that ended up being a very, very complicated story.
Okay. And again, to catch the listeners up, you're saying that the character trait that you noticed was the ones that were CIP positive, the ones that could metabolize the citrate. And the part of the contingency experiment showed that there had to have been some previous mutations, other mutations that you can't, at this point, you don't know what they are. They could be anything, but those had to be in place before this subsequent mutation made it CIP positive.
Right. So I knew that whatever they were, they made it easier to evolve CIP+. Evolving CIP+ went from a rate of vanishingly low, so low that I never saw an instance in the ancestor, to about 1 in a trillion in later generations. But as far as what those potentiating mutations were, we had no idea. And my PhD ended up taking extra time because I was convinced maybe we just sequence a few more genomes and I'll figure it out. And that wasn't the case. A couple of Jeff Barrick's graduate students came in later to work out that story as part of their PhDs. So I was on a fool's errand, but in science, you don't know how difficult a problem is until you start working on it. And what they uncovered was really very interesting. And it wasn't simply that mutations arose and created a genetic background in which CIP+ could occur. It was more complicated. And it goes to ecological changes that occurred.
Okay.
Yeah, and this is a story that we've seen in a number of the populations. So to give you the outline, early on, mutations arose that improved ability to grow on glucose and made it so that the cells could just start wolfing down glucose, but at the price of excreting large amounts of acetate, which is the acid in vinegar. So they ate glucose, they pooped out acetate. The acetate was a new food source in the environment. And in due course, cells evolved from within the population that were specialized on the acetate. And they picked up mutations that improved their ability to grow on the acetate. And they picked up more mutations that improved their ability to grow on acetate. And eventually a mutation occurred in a gene called GILT-A, which encodes an enzyme called citrate synthase. And this convert a chemical called acetyl coenzyme A, which is what glucose is turned into by glycolysis, which some of your listeners may remember memorizing and forgetting multiple times like I did in college. And the thing about this mutation is it improved carbon flow into what's called the tricarboxylic acid pathway that is needed to metabolize acid. This is important because it made the cells better at using acetate and coincidentally better at using citrate before the ability to grow on citrate had evolved.
Okay.
What Eric Quant, the grad student who figured this out, discovered was once this mutation in GILT-A was in place, the ability to grow on citrate became slightly beneficial. So if you made a mutant with this mutation and you made it able to use citrate, it was slightly beneficial. The citrate users had a fitness benefit of about 1%. But if you take this GILT-A mutation away, access to citrate is actually toxic. So this adaptation to acetate that was produced by improved ability to use glucose set the stage for final mutation to use this citrate. And without the sequence of events, it wouldn't have happened. And a later grad student, Dasha Leon, showed that another nifty thing here was all of this came together right when the supply of mutations with benefits higher than 1% had been exhausted. And had CIT plus arisen in this background prior to that point, it would have been outcompeted in the population by these more fit mutants.
Okay.
So it just came along coincidentally at just the right time after this specific sequence, and the new trait was there. And once it was there, natural selection refined it.
Right.
Right.
Such a more nuanced story than just simply talking about breaking genes, I've got to say.
Exactly.
Zach, the reason I want to bring you back 5 minutes ago, this what you described as there had to have been a number of mutations that were invisible to you and your researchers. Clearly, the evidence after the fact showed that these changes had occurred, but there was no way to know that they were there until finally something else happened. Another mutation occurred, and now the cells became CIT positive and they were able to outcompete their cousins. So a number of invisible changes had to occur before you saw this new innovation. And that's very different than the way creationists and the ID proponents will often portray the situation. They will say, look, you start off with a gene that works really well, and then you change it. All you're going to do is make it work less well. And the more changes you introduce, you're just going to make it less and less well. You're not going to suddenly make it work into something new. That's what they say, that can't happen. But you're showing a mechanism where you can have these background changes that were invisible. The cells still continued to grow. They weren't growing less well. And then when the combinations were right, now you had a population of cells that suddenly grew better. A whole new line that can grow on citrate.
Right. So the reality is what this shows is that you can have these changes that are rejiggering, to use a technical term, improve metabolism to certain conditions. In this case, growth on acetate. And those changes not only do improve things, yeah, it makes them worse at growing on glucose, but better at growing on acetate. It goes back to the whole jack of all trades, master of none thing. There are trade-offs. And evolutionary theory predicts that. That actually is something that really gets me about some of the intelligent design creationist critiques, which is, well, you change it, it's worse.
Right.
It at the original thing, but it's better at the new thing, but it's worse. Okay, that's not what evolutionary theory says should happen. That is an issue that I have with their arguments. It's because they very rarely engage the actual science. And they argue that what we see disproves evolution because they say that evolution should show things that evolutionary theory doesn't agree with. So they are creating a straw man of evolutionary theory and criticizing our data from that perspective, even though it doesn't actually engage with the actual theory.
Right, okay.
We expect, as evolutionary biologists, to see during adaptation to particular conditions, changes that improve the ability to grow under these conditions that may have the cost of reducing ability to grow under other conditions. That's just what we expect from the theory. And when intelligent design creationists and other creationists criticize, they're not criticizing from the standpoint of the actual science.
Right.
Yeah. And that always really bothers me because it's not scientific criticism. And these are people who are sophisticated enough to know it. And that they do this has been pointed out many, many times. And yet the arguments never change. Behe makes the same criticisms of evolutionary theory that he's made for 30 years, despite it being pointed out that many times he's not engaging the science at all, that he's outright wrong.
Right.
And you just see different iterations in response to findings that we have. And that's been true definitely with the citrate story.
Okay, so Zach, why don't we move more into this citrate story? You've already told us how there are genetic changes in citrate uptake, citrate metabolism, but you didn't explain to us how the cells are better to grow on citrate.
Yeah, so something that I realized while hiking one day is that the origin of a novel trait, like the citrate users in this population, are going to have 3 steps. One is potentiation, which makes it possible to evolve this new capacity. Actualization, which is a final genetic change that creates or instantiates the trait. And then usually when that happens, the trait is not very good. It's weak. And then you have refinement where mutations accumulate that improve. So I've talked about the potentiating phase. The actualizing phase was a final mutation that we figured out was a duplication of a stretch of DNA that was 1,733 base pairs long. And it overlapped that sit operon that I talked about earlier that encodes the genes that are needed for anaerobic growth on citrate, fermentative growth on citrate when oxygen is present. And what was beneficial about this duplication is it contained the entire gene that's called CYP. CYP encodes a transporter for bringing citrate into the cell. It's the transporter that's used to feed on citrate when during anaerobic growth on citrate. And it also was in such an orientation that it placed this copy of this new second copy of the CYP gene under the control of a promoter, which is a regulatory region that tells the cell when to turn a gene on or off, that normally controls a nearby gene called RNK. RNK is turned on under conditions when oxygen is present when food runs out. So this promoter for RNK is now controlling CYP and turns the citrate transporter gene on under conditions containing oxygen when glucose runs out. And this means that these bacteria now have the ability to uptake citrate from the environment and feed on it. This is what caused the citrate. Now, what's really beneficial about this mutation that's often misrepresented, especially by critics, is that it was just a matter of breaking the regulation gene. And that's not true. It's not a matter of breaking regulation. The way this duplication occurred, it's tandem and head to tail. So you have one copy right next to another copy. And what this ends up doing is that the sit operon is still intact and under its original regulation. The RNK gene is intact and still under its original regulation. But in between those two, you have this new regulatory module made up of the RNK promoter, a copy of the RNK promoter, and a copy of the CYP gene. So you have a new regulatory unit without breaking it. That's new information that's made out of pieces from the old without disrupting it. You lose nothing, but you gain something. And this is what leads to the production of the citrate transporter when oxygen is present.
Okay.
Now, one of the really amazing things is I mentioned a while back doing all of these replay experiments when I found 21 independent instances of the evolution of syphilis. So I analyzed the genetic changes underlying them to look at the actualizing mutation for them. And in each case, it was a different mutation. So there were a number of duplications that were similar to the one that happened in the main population, were slightly different. Sometimes a different promoter was co-opted. In one case, there was a deletion that created a new promoter whole cloth. There was an instance where the DNA inverted and put the copy of CYP under the control of another promoter. And then there were a number of instances where copies of an insertion sequence called IS3 inserted adjacent to the CYP gene. And IS3 is very interesting because it has promoters in its end. And so IS3 inserting there turned on CYP. So this final mutation could have happened in a bunch of different ways. And in each case, it involves activation of this CYP, this pre-existing transporter, which, oftentimes intelligent design creationists and others will criticize this, saying that, well, this didn't really evolve because it was already there. But evolution of new traits often occurs by what's referred to as evolutionary tinkering. This is a term that was formulated by Francois Jacob, who was a Nobel Prize-winning molecular biologist, He wrote a paper called Evolutionary Tinkering in the 1970s. And he pointed out that evolution is not like an engineer, just creating things anew for different purposes. Instead, evolution is like a tinkerer. You have mutation, just trying different variants of things, putting things next to each other, like a tinkerer does in a junkyard, just playing with things, seeing what connects. And every now and then, those new combinations will have some function. And it may not be a very good function, but once it's there, it can be refined to have a better version of that.
Right. Okay.
It's making use of what's already there. And so that's exactly what we would expect from it. This is how innovation often occurs, is recombination of pre-existing things. As Jacob put it, to create is to recombine.
Okay.
Yeah.
So just to summarize then, and we covered a lot of ground there, a lot of names and technical jargon, but basically you said what you've learned from this is then you have this 3 steps in evolution, that you have what you call potentiation, which is basically some changes that make things ready for a new trait or character, a phenotype, yes. But it's just basically a new function. Let's call it that.
Sure.
Potentiation is getting it ready for a new function. The second step is actualization, which is now the function is there, but it's not so great, but it's there. And then the third step is this enhancement, where it's refined, it's made better.
Right.
And all of this involves some minor changes, progressive accumulation of minor changes to the DNA. You talked about insertion of sequences between 2 genes that were still functional, but insertion of a couple new pieces of sequences that just fit together in a certain way that they now turned on sit all the time instead of waiting for the oxygen to be absent.
Right. Not turning it on all the time, but specifically in the case that evolved in the era-3 population, it turns on the citrate transporter when oxygen is present and when glucose runs out.
Okay.
Yeah.
So that's a lot of genetic changes and a lot of functional changes. The creationists will talk about, for example, we had Michael Behe on, and he said, you had 75,000 generations. Did you see a whole new species? Did you see new organs? Well, he specifically referred to new nanomachines. Let's talk a little bit about if that's even realistic. What does 75,000 generations work out to in human time?
So to get in human terms, approximately, you need to multiply 75,000 by 20.
20 years, right?
Yeah, if you assume 20 years is a standard human generation, as opposed to the E. coli that double every 20 minutes or so.
So 75,000 generations is only, in air quotes, a couple million years. And if you take into account, for example, the fact that chimps and humans diverged 6 million years ago. Now, chimps and humans are still very similar organisms from a functional, from a physiological point of view, and in many ways from a genetic point of view. They're very similar organisms, and yet it took 6 million years for those two to evolve. If you talk about humans replacing the dinosaurs, like how far back do we have to go? Well, you would require 100 times as much time, 100 million years, not just 2 million years, which is what your long-term evolution experiment represents. It's only 2 million years of, we'll call it human time.
I hear that criticism that Behe and other intelligent design creationists make, and it doesn't make sense to me because
exactly
no one would predict wholesale new molecular machines to evolve de novo. And that seems to be what he's demanding, that new molecular machines evolve de novo, ex nihilo, from nothing. And
right.
Evolutionary biologists wouldn't expect that. That's not what the theory predicts. Again, we expect, especially in the conditions that we're looking at, the only supply of variation is what is being generated by mutation of what already exists in the genome. And I think of evolution as being very clever because it's natural selection's working on randomly generated variation. It has no preconceived notions. It just tries whatever occurs. So it's the cleverness of mindlessness, but it's also lazy. And if we gave it another novel resource to grow on, you would expect, if it's possible to grow on it by just recombining what's already there, that's what's going to happen.
Right.
That's the variation that's more likely to arise.
Okay.
Yeah, just creating something whole cloth is very unlikely over the time period that we're looking at.
Right. So it's totally unrealistic.
Right.
You mentioned how evolution is, you talked about clever and tricky or nifty, I forget the word that you used. But the other thing that needs to be said, needs to be acknowledged, is that evolution may not necessarily be elegant. It doesn't always come to really elegant design. Sometimes they're very bizarre ones.
Yes, exactly. Sometimes evolution can produce elegant results and you see elegant adaptations, but yeah, so often you don't, because evolution jury rigs. Like I said, with evolutionary tinkering, it's building new things out of old things, and so
Right.
you have all sorts of compromises and weird designs because it's not building things anew; it's modifying what is already there in most instances. You have instances like the recurrent laryngeal nerve, I think it's called, that
Right.
in us it goes, it innervates, I think, part of the jaw, but it comes down into the chest around, I think, the aorta and comes back up. It's a stupid
Yes.
design if you're starting out from scratch. But if you're just making small changes to something that already exists, you end up with things like that, just because that is how you get to a new place. It's just these tiny tweaks that sometimes end up working, and they work well enough. They don't work perfectly, and the design isn't perfect.
I'm going to come back to laryngeal nerve. You did a great job explaining it, but it was a thumbnail sketch, and I want to give a bit more detail. This is an example of what we've talked about in the past in previous episodes about clumsy or awkward design, which, you can wave your hand and say, okay, well, what difference does it make? But it just speaks against the idea of this having been designed, as opposed to another set of designs that we refer to as malevolent, and that clearly speaks against a good designer, a benevolent designer. So here's an example of a nerve that comes down from the brain. It innervates your voice box, but it first comes down from the brain, goes down to the heart, loops around the aorta, and comes all the way back up to your throat. And you look at that and you think, that's weird. Why would that happen? And if you take it a step further and look at giraffes, which have a very long neck, they've got this nerve that travels, I think it's 12 feet from the brain down to the heart and back up to the throat. And it just makes absolutely no sense until you go back through evolutionary history, look at where this recurrent laryngeal nerve came from. When you go backwards through the tree of life and the animals that evolved from each other, we come back to a period where you have animals which were essentially fish, not the way we see fish today, but the ancestor of the fish, where the brain and the voice box were like almost neighbors, and the heart was a little bit in front of that. All right, so you got the brain connecting directly to this part of the body that was their version of a throat, and a heart right beside that. And as this animal began to evolve, and the head moved up and the body went down, and the heart moved over, this nerve became longer and longer and longer and longer, until, in the case of the giraffe, it goes 24 feet in the wrong direction. So, perfect example of what I call a clunky design that does not speak towards a designer.
This is exactly what you would expect. And,
Yes.
something that Darwin wrote about way back when and is often commented on now is some of the greatest evidence for natural selection is not perfect adaptations, but imperfect adaptations. Because
Right.
sometimes you just can't get there from here.
Right.
Yeah. Oh,
so
I will note just there, one premier example of that is an enzyme called rubisco that catalyzes an incredibly important step in photosynthesis. Most of the biosphere on Earth depends on the activity of this enzyme. And the thing is, it originally evolved when there was very little oxygen in the atmosphere. And now there's lots of oxygen there. So in the presence of oxygen, it does not do its job effectively. And half the time it produces a product that has to be dealt with and takes up a lot of energy to do it. But there's no way to fix it because it's so key. It's a frozen accident that's there, and everything else has been built on top of it. So that's another instance.
Okay, so Zach, we're getting a little bit short on time. I think we should finish off the story of the citrate metabolism and what you found as part of this LTE team.
Well, one thing that I can say with regard to that builds on what we were talking about earlier with stupid design is the use of the CYP citrate transporter is actually an example of stupid design. It's very much a jury rig. So this is a transporter that is adapted to use under anaerobic conditions. So citrate fermentation brings citrate in and converts it to succinate. Well, CIT pumps succinate out into the medium to bring citrate in. It works well in that instance. But here, what it ends up meaning is that citrate's pumped into the cell using CIT while it pumps succinate out of the cell. And succinate is an intermediate in the biochemical pathway for making energy from citrate. So the way I describe it is with this transporter, it's a little like a baby learning to eat, where it gets food into its mouth and then a lot of it dribbles right out.
Okay.
And that it's very compromised. And the earliest CIT plus cells were very bad at using citrate because this is such a junky design. In order to correct it, there has to be another mutation that occurs that activates expression of another transporter called DCTA. And this is a transporter that brings the succinate back into the cell. So essentially, citrate goes in through CIT into the cell. It gets converted to succinate. The succinate goes out of the cell. Then it's brought back into the cell by DCTA. And it's just a remarkable jury rig that makes no sense. But it's something that's necessary because this is what evolution has to work with here. It's building the new from what it has. It's a stupid, clunky Rube Goldberg design, but it's what's there.
It works, yeah.
Yeah, and that's true of what we see all across evolution and across all of life. Now, that mutation that causes expression of DCTA, that's an example of these refining mutations that I mentioned earlier that improve this new trait and alter metabolism and physiology for growth on citrate. And the thing about the refinement phase is it's open-ended. There are a huge number of mutations that can improve the trait. And that's still going on. And that has a neat consequence. So something that I have found is over time, the citrate users are getting more and more fit on the citrate and succinate. This two-part niche that they're now in. They're getting better over time. At the same time, they're getting worse on glucose, on that ancestral resource. They're becoming ecologically specialized. This is exactly what we expect to see with new species. It's diverging ecologically from its ancestors. And so we are seeing speciation occur in the lab in this population. And one of the really amazing things about that divergent fitness trajectory on the citrate and the glucose is they correspond to each other, which indicates that the same mutations that are refining the ability to grow on citrate and succinate are reducing the ability to grow on glucose. And we're going back to jack of all trades, master of none, trade-off.
Right.
And I have a mentee now, Claire Spender, who their job is going to be finding candidate mutations that are underlying this and testing them by moving them into different genetic backgrounds to see if they improve fitness on citrate, but also reduce fitness on glucose. And when they're done, that work will be really remarkable, showing us the genetic basis of incipient speciation.
And to bring back that analogy that I used earlier about the marathon runner, you're talking about bugs that are getting better at growing on citrate and succinate and not as good on glucose, just like this marathon runner who's getting better and better and better at running in the heat, as opposed to the guy standing at the bus stop who's got his park on and he's got 10 pounds, he's 10 pounds overweight. Put those two again in Alaska, the marathon runner is just not going to be able to grow in Alaska.
Yeah, I would go back to the pole vaulter and the marathoner. That as the person practices the marathon, they get less good at pole vaulting.
So two diverging athletes, or as you put it, two diverging species.
Exactly. And the really amazing thing about the long-term experiment is things like this arise. I say sometimes that the long-term experiment, the LTEE, is a model system that produces new model systems. And here with CIT+, we have speciation occurring in front of us in the lab so we can use it as a model to understand speciation.
Right.
Now, relatedly, and going back to contingency, is one of the other things that we can do in the system is, I'm sure that you and many of the listeners have played the what if game. What if Hitler had died in World War One? Or, what if I had turned left? And so, what if Hitler died in World War One? Because he did fight on the front lines there. He could have been killed, in which case, who knows what would have happened.
Okay.
Yeah, or what if the South had won the Civil War? These are perennial ones. We have them about our own life. What if I hadn't met my spouse? And the thing is, these are always thought experiments. But we can actually make them real in an experimental evolution context because we can access different points in history with the frozen fossil record, those frozen samples at different time points in the population. And we can go back and make changes and then see what happens. It shows how just every time we look at something, it yields new questions, new phenomena.
Right.
It's fractal.
Yeah.
It just keeps getting larger and more interesting.
Exactly.
Yeah.
And I appreciate that because I've done research as well. It's such a fun game. But we need to draw a line under this conversation. Maybe we'll maybe you and I should talk about how we can develop a third episode.
I'd love to.
But is there any remaining things that really need to be said? And otherwise, then I want to hear about what is the future looking like for you? Are you any books in the works? Any speaking engagements?
Not immediately. There are books that I would like to write at some point. I don't want to commit myself to anything right now. I'm always willing to speak if someone invites me because this is just spectacular stuff and I love talking about it. I'm continuing to work with Rich here at MSU on these aspects of the experiment that I've talked about. Like I said, there's this work I'm doing with Claire Spender. I have another mentee, Max Halliday, who I'm working with a colleague at the Cleveland Clinic to look at the evolution of antibiotic resistance in this population. And beyond that, as you noted in my intro, I'm now the Director of Graduate Education Innovation in the Department of Microbiology. And I'm going to be redeveloping the graduate curriculum for microbiology students going forward to emphasize the philosophy of science, the logic of science, how to think like a scientist. And I really think that that's where my major contribution to science is going to be, helping mentor new students in new ways of thinking in order to be more effective scientists. And I'm really looking forward to that. But I'm still going to keep a hand in the lab. I'm still fascinated by contingency, and I'm working with some philosophers on aspects of that. So the future is bright, and I wake up every day amazed at my good fortune that I ended up having a path that brought me to MSU and to this lab and getting to work under someone like Rich, who is phenomenally brilliant, but also an incredibly nice person. He's just a golden human being. And I'm very, very lucky to get to work with him in this lab and with everyone I've met on this project. Like I said, there are dozens, if not hundreds of people working on it. And every single one is a really great human being who works incredibly hard to untangle these mysteries of nature that we're finding in these little facts.
Excellent. Well, keep up that good work. And we're going to keep you on speed dial because I think we're bringing you on again for some follow-ups. So thanks for sharing your time with us, Zach. We really appreciate it.
Thank you. It's been wonderful. Thank you so much for this opportunity.
Okay. And we will talk again. Bye for now.
Bye.
So, Scott, this episode was hard. What did you get out of it? What questions do you have about what Zach said?
Yeah, well, first, I found it very fascinating. Secondly, it raises a whole bunch of questions in my mind that probably aren't answerable, scientific questions, which is great. That's what good science does, right? And I'm sure that people working on the project in the lab have many more questions now that they've learned some things about this. One of the things, does each change need to confer a benefit, or do some of them lay latent and then later confer a benefit, that kind of thing?
Right.
Yeah, those kinds of things. So Yeah, it was definitely, for me at least, it was it was a science, of course, it was a scientific discussion. If you're not interested in science, if evolution doesn't matter to you in any way, then that's fine, and maybe it isn't. As we mentioned later on, we are going to talk about more the philosophical implications, the religious implications of some of these things, and hopefully this will have provided some foundation for that.
And by later on, you mean next week, Scott?
Yeah,
So
that's right. That's right.
as for myself, I come away from this interview with Zach with mixed feelings. On the one hand, I was amazed at the science. It was so satisfying to see the pieces falling into place and how we're getting more and more detail in how evolution produces the biological diversity that we see all around us. And I really appreciated it, really resonated with Zach's enthusiasm at the end of that conversation where he said every day he counts himself lucky to be doing such fun work with such great people. But on the other hand, I'm once again really disappointed with how so many Christians have responded to this. Their acceptance of vague, hand-waving worldviews from non-experts and dismissal of detailed explanations based on actual data presented by the experts, their promotion of blatant lies and half-truths and misrepresentations. I'll even say their short-sightedness and their closed-mindedness. So last week we said quite a bit about the leaders, and I'm not going to get to talk about them in particular again, but I want to talk to the community instead, that we need to be a bit more careful about who we're putting on the stage, who we're putting on the podium with the scientific experts, the ones who actually are doing the science, who have the training, who have the experience. I'm sorry if this comes across as too confrontational, but let's face it, if you needed surgery or the transmission in your car replaced, or have a cake made for your daughter's wedding, or even you needed a haircut, would you go to somebody who's only read about those things or someone who actually has hands-on experience and does it all the time? And so I ask, why do you, the community, put them on the pedestal beside people who actually do the work, who have problem-solved their way through it for decades, and who have published their findings after critical review from their peers? And the only reason I can see is that their message is what the people want to hear. It's confirmation bias. What do you think, Scott?
Yeah, to me, it's very obviously confirmation bias. I think the ID movement kind of, and I can say this for myself, when I first saw Behe's books and the books on the design inference and so on, I thought, oh boy, these are substantial arguments. These are, like Dr. Behe is a biochemist, and the design inference is a published peer-reviewed thesis by, I think, Oxford University Press or whoever. So all of a sudden, it seemed to put the idea of design as a viable, an intellectually respectable option. And so that attracted me. That's my confirmation bias, right? I'm a theist, so, I was attracted to that. At the end of the day, however, unfortunately, I believe, as we've talked about, it turns out that isn't the case. And so we have to be intellectually honest, I think, as evangelicals. Because if I talk to many of my evangelical friends and on this subject, they'll have read Behe, they'll have read various, but they will not have read the other side at all. And when we interviewed Frankie Schaefer, he made a point that I thought was really right on. That his father, it's kind of like his father would go read all of these secular philosophers and so on, and come back and tell evangelicals what they should think about them. And that goes on a lot in our churches and in our community, where we have gatekeepers who go and they study these people, and then they come back and they filter it for us. But I think if we want to be part of the modern conversation, we need to be people who, A, are honest about truth and are willing to seek it wherever it leads, and B, make sure that we are not, just like you said, confirming our own bias. And in the day and age we live in, we know this is a massive problem now with the proliferation of information through the internet and so on. So
right.
I think it's more than ever, the most important thing to teach children, I think nowadays, more than content, is how to think,
how to discriminate,
just how to discriminate, how to think. That's the most dangerous thing I think that we may be missing, because if we don't do that, then the danger is we're going to end up, the danger is we're going to kind of end up where we are, right? Where we have a proliferation and by the way, on both the left and the right, it's not just a right-wing thing. That's the more That's the more kind of maybe obvious one, or the one that gets more press. But no, there's plenty of it on the left too, and yeah, it's leading us to kind of a societal madness that it may where everybody's worried about artificial intelligence. I'm just worried about human intelligence right now.
Yeah.
Anyway.
The last thing I do want to say then is I am sensitive to the fact that there may be listeners who think, look, Luke, all you're doing is taking away things that I can believe in or that I once believed in. You've taken away, my belief in a literal Bible. You've taken away young earth creationism. Now you're taking away intelligent design. All you're doing is taking away. I don't think that just because we find the flaws in some of these ideas and get rid of them, that means I have to give up my whole entire faith system. Just because I'm left with questions that now don't have convenient answers, that's not a reason for me to continue to hold on to worldviews that aren't supported by good data and good logic, and in some cases, quite frankly, they're supported by lies. I'm sorry. The whole counter-argument that the LTEE project has only shown how to break genes rather than build up genetic information is quite frankly a bald lie. On my main blog site, I have a quote that's been credited to Carl Sagan. It is far better to grasp the universe as it really is than to persist in delusion, however satisfying and reassuring.
Amen.
Okay, I get it. Enough said. Scott, what are we going to do next week?
Yeah, well, next week we're going to unravel another one of the loose threads that was left hanging after our deep dive into intelligent design. We're going to talk to someone who's making a name for himself in the scholarly area of, ready for it, teleology. There's a $1 million word. And all it really, means is teleology is the idea that things have purpose, things have an end. So the idea, for example, that a watch is made to tell time, right? That has a purpose, it has an end in mind. And we're going to talk as well to two world-leading philosophers who have written and spoken extensively on this topic. The basic question we'll be looking at is, why do biologists find it basically impossible to talk about evolution without using language that implies purpose, intent, or meaning? And in this very discussion we've been having, Luke, it's been pretty obvious that we've used a lot of language like that. It's just very difficult to avoid. Statements like fish have fins so that they can swim better. Viruses and bacteria mutate so that they can infect you better. Birds fly south in order to escape the winter and find better food reserves. Interestingly enough, I spent the last week banding owls out here in Victoria that are doing that very thing right now, heading to places where they can get more food. And we do talk about it that way. Natural selection wants to increase survival and/or reproduction. You won't want to miss this, and we'll see you next week.
Bye for now. You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.